Welcome everyone. So I don't know if you guys know, but today is actually a really special day in science and specifically in chemistry because today is what's called Mole Day. And so I wanted to talk to you guys a little bit about what Mole Day is. I'm a chemist, so this is a really special day for me. But we wanted to talk a little bit about what this really means. So this is just a quick little introductory talk while a few more people are still joining us. Okay, so before we can really talk about what a mole is, we have to understand these different names that are associated with like amounts, so different like ways to count. And so some examples that I thought of is when we talk about a dozen, this always means 12, right? Okay, Elijah's way ahead of us. He already knows what a mole is. But a dozen, when we say a dozen, we mean 12 of something, right? When we say a pair, we mean 2 of something, like a pair of shoes or a pair of socks, something like that, right? Okay, can anyone think of any other examples of things maybe that come in like a certain number that always mean a certain number? Triplet, that's what I was thinking. That's really good. A trio, yeah. Quad is 4, very good. Half a dozen is 6, good. Couple, trinity, these are quartet. Oh my goodness. Okay, so you guys totally get this idea. These are fantastic. So these are just numbers that we use to mean, or words that we use to mean a certain number. It's kind of like shorthand. And so when we say a dozen, everyone knows we mean 12. And so when we say a mole, we mean this really big number called Avogadro's number, which is 6.022 times 10 to the 23rd. And so the actual definition of this comes from the number of atoms in 12 grams of carbon. But it's just kind of like a fun number to think of. And this 10 to the 23rd is why today is Mole Day, because it's 10/23, right? October 23rd. Yeah, it's written in scientific notation. Nice. Yeah, you guys got this. So yeah, so Mole Day every year is celebrated on October 23rd. So we're really excited to be doing science with you this year on Mole Day. Super fun. And I just wanted to do a couple fun facts about really how much a mole is, because I think really big numbers are sometimes really hard to understand. So we already said it's defined based on the number of carbon atoms in 12 grams of carbon. 1 mole of water is 18 grams or 18 milliliters, which is like 1.2 tablespoons. But if we're thinking about big things, like 1 mole of donuts would cover the whole Earth like 5 miles deep. Yeah. 1 mole of basketballs is about like the size of the Earth, which is obviously really big. I thought this was kind of fun. If you receive 1 mole of pennies on the day you're born and you spend $1 million every single second until you die, you'll still have pretty much all of your money left in the bank when you die. So a mole is a really, really big number. And so I just wanted to say happy Mole Day. And we're really excited to be here doing science with you on this really special day. So I'm going to stop sharing now and hand the floor over to Danny to really introduce you to what we're up to today.
Bienvenidos. Welcome to this amazing program. We want to thank Yale. We want to thank ARTE and our board for this amazing opportunity celebrating Hispanic Heritage Month with amazing science project. So I'm going to cut the ribbon and start the program. Let's begin. Take care. Enjoy it.
All right. Thank you so much for that introduction. Today, we're going to do some really exciting science demos. But first, we have a really exciting guest speaker for you all. And so today, we're going to have Dr. Luisa Escobar-Hoyos talk to us a little bit about her pathway into being a scientist. So, Dr. Luisa Escobar-Hoyos is currently an assistant professor in the Department of Therapeutic Radiology at Yale. And there she studies RNA processing in the context of lung and pancreatic cancer, with the goal to find better therapeutics for those two cancer types. So she does really exciting research, but today she's going to tell us something probably even more inspiring, which was her pathway into becoming a scientist. So with that, I'm going to have Luisa give a talk for us today.
Buenos dias, everyone. Good morning. I hope you're well. Thank you so much for everyone for being here, for the parents, for the kids, for getting up early to hear a little bit about, all of these great program that is prepared today. And what I'm going to do is I'm going to tell you how I decided to become a scientist, what was inspiring in my life that brought me here. So I want to share a couple of slides that I put together. So today, as I was mentioning, I want to share with you a little bit on my path to becoming a cancer scientist, starting as a professor in Yale a couple of years ago. And my big message today is that you need to dream since you're little. I know you guys are 9, 12 years old, and you might be thinking already, what do I want to be? Do I want to study physics? Do I want to study science, biology? Do I like a little bit of both, as we were seeing in the chat? So I hope that, the story that I'm going to tell you inspires you to become whatever you want, because you can do it and you have everything to reach it. You just need to be persevering. So I was born and raised in Colombia, South America. So for those of you who don't know where Colombia is, it's actually at the north in South America. So it's located here in this corner, kind of limiting with Central America. And this red dot here in the southwest of the country, that's where I was born. I was born in a very small town called Popayan. It's very, very tiny. A lot of people do not know it. But I wanted to give you and tell you that I come from this very small town to give you a sense of all of the path that I had to kind of go through my life in order to be here now in the States. And I've been here for the last 10 years. So how did I become a scientist? When was it that spark of wanting to learn about science became? So this is a very personal story. I was actually inspired because both of my parents, one is a mathematician and the other one, and my mom, is a biologist. So at home, we had a lot of kind of science related conversation, mathematics, physics conversations at the table. And that was very exciting to me since I was a little girl, because I could ask, pretty much a lot of questions at home and try to get the answers from my parents. My mom is a biologist. And I remember when I was about your age, I was feeling sick at school and I had to go to my house, but my mom said, I'm going to pick you up. I'm going to bring you to the laboratory, because she also has a laboratory. And I need you to wait here until I finish my class. And I said, sure, not a problem. I'm feeling better. And I was looking through the door and I was seeing how she was teaching this group of like 20 students, and all of the students seemed very happy. They were asking her a lot of questions. They seemed very motivated about science. And that really, touched me. I had seen my mom teach us to my sisters and me, but I had not seen how other people were excited when she was teaching them and how all the students were learning from her. So this is my mom. I wanted to show you, we look alike. And she is my pride, she's my role model. And this is in Spanish, but this is a quote from my mom. She says, since I was very young, I wanted to be a mom. I wanted probably to follow the good example that my mom left me. It's been one of my biggest commitments to bring my kids to be professionals. And that's why I feel so proud. And that's my mom. And this was a quote that that she said not too long ago. But, reflecting on this quote, I am now a mom, and I also want my kids to be, whatever they want to be, and I want to support them. So I want to, call out there to all the parents that are hearing me to know how important it is to support your children for whatever they want to be. It really starts at home when you support their questions, when you tell them, you give them wings to dream of whatever they want to be, because they can, because each one of you has all the tools and all the capacities and to be inspired and to become whatever you want to be. So how did I come to the United States? Since I was in Popayan, we were a middle-class family. We didn't have a lot of money to travel but we did had a little bit way to, to study. So I want to tell you a little bit about what was my path and what did I learn through my path. So after I decided, and by the inspiration of my mom, I wanted to become a cancer scientist. I wanted to potentially change the way of the course of this disease. As many of you know, cancer is right now a disease that kills hundreds of people in the world, unfortunately. What we don't know is that cancer is not just one single disease. It can be up to 800 diseases. So we're actually fighting across multiple cancer types, and we need to understand them better. So when after I graduated from high school, I went to college to study molecular biology. And for me, that part, when I was an undergrad, I did my undergrad back in Colombia. And during my undergrad, while studying biology at this university in Colombia, it was really important for me to start finding my mentors. Who could I seek help from? Because I was now in this city, different from my mom, and I was by myself trying to kind of learn the way about how undergrad was it? How could I find professors that could help me advance my research? And I had a really nice professor who encouraged me to participate in a symposium in the university. And during that symposium, they had invited speakers from the US, from different universities. And at that time, I got to meet two professors, one from the University of Illinois and another professor from Columbia University. And it was because I was so excited about, learning and potentially doing a research fellowship in the US. I met them and I started just talking to them in one of the breakfasts during the meeting and just saying how excited I was about, becoming a scientist, a cancer scientist in particular. And they were so nice and so kind that they invited me to do a summer rotation in their labs here in the US. So during a couple of years, I was able to go into my first summer internship at Illinois, and the second summer internship the year after at Columbia University. And in all of these experiences, I was trying to learn, how do cancer forms? Are there any particular factors in the environment or in the food or at our jobs that can potentially increase the risk of cancer? Because my thought was that potentially we could decrease the number of cancer cases by preventing them. But my biggest learning experience from this is what I have highlighted here: the importance to find your mentors, the importance to find people that can help you in your career. Most of the time, we're not going to have people that are going to come to us and say, how can I help you? But we can go and find them, and they would be very encouraged to, once we find them, to get their help. So don't think that if people are not coming to you, that you cannot go to them and ask for help. Then I was doing my master's after graduate school, because I still wanted to kind of refine a little bit about my scientific training and really decide if I want to become and commit to being a cancer scientist before going to the long, PhD training, which is 5 years. And so during the master's time, I started preparing. I decided I did want to train as a scientist and conduct my PhD studies. But at that time, I was, trying to study really hard, because as an international, not having English as my, my native language, and also having trained in another country, I needed to take a lot of tests to make sure that I could prove myself that I was good enough to come to the US. And I have to say, this was one of the most challenging times of my life, because I had to take these exams over and this test, sorry, over and over again, because they were really hard. They were tough. And I remember one day crying and telling my mom, I don't think I'm going to be able to do this. This is really hard. And, you know, never, ever she or my parents ever doubted of my capacities. And they just helped me to try find ways to get the books, or to find better areas where to study, or even to help me with, you know, with my food and with other, you know, things that they could provide. And after 2 years of trying really hard to pass these exams and get the scores that I needed, I was able to get, you know, to get them. And so the second message is to don't give up. Please don't give up. Find different ways. It's okay if you're tired and you want to, you know, spend some time in thinking about it. Then you can come back and try again. It doesn't hurt to try and to give your best. So gladly, after, you know, taking these exams and finally, you know, getting the right score, I was able to come to the US to conduct my PhD studies. And so I trained at the University of Stony Brook, which is just across from it's in Long Island. So from here, from the south of Connecticut, we see the Long Island Sound, and to the other side there is a small island, and that's where I went to graduate school. And during graduate school, what I learned is that I needed to take every opportunity. If there was a class where they were teaching how to better speak English, I would go to that. If there was a class on teaching or any activity on how to train your skills in the lab, I would sign up for that, because I wanted to learn as much as I can and make the most out of this opportunity that I was having to be here in the US and learning. So the PhD process or training takes about 5 years. So basically here is where you go to the university to train how to become a scientist, how to go from asking a question of interest and coming up with a whole methodology and later testing it in the lab. So this applies to medicine, this applies to physics, this applies to chemistry, as we were just hearing. So when you decide to be a scientist in your area, most likely you would be going to a PhD training, and it's a lot of fun. It was one of the best learning experiences in my life. So afterwards, and after I did very well at Stony Brook, I wanted to do, it's an intermediate kind of step of training between the PhD and becoming a professor, which is called the postdoctoral training. And at this time, I wanted to go into a cancer hospital to train a little bit more on a new disease, because it turns out that when I was a graduate student, a PhD student, I was studying one cancer type, but now I wanted to study another cancer type. And in particular, I wanted to study pancreatic cancer. The reason why I wanted to study this disease is because it's a very lethal disease. It's a very hard disease to cure. And so I wanted to bring all of my knowledge and all of my energy to really devote to this cancer type and try to understand it and trying to find its Achilles heel. So after training in pancreatic cancer in New York City at this hospital called Memorial Sloan Kettering Cancer Center, I came here to Yale a couple of years ago, soon before COVID, the lockdown started. And I started my own lab to study pancreatic cancer, and now we're expanding to lung cancer. And what I wanted to say is that at this time, now my biggest commitment, I have my lab for two main reasons. The first one is, as I was saying, I want to try to find a cure for pancreatic cancer. But the second reason is because I want to train the next generation of scientists. I want to be like my mom, inspiring other people to become scientists, inspiring people to love science or to love whatever they do. And so right now I have a team of about 10 people, women and men at different stages. So some students are undergraduate students, other ones are graduate students, so doing their PhD or their master's, and others are at the postdoctoral training. And we have people from different backgrounds, Hispanic, Asian, American, and it's a lot of fun that we get to conduct science as a team. So I don't know how we're doing in time, but if I have maybe a couple of minutes, I just want to tell everyone a little bit on the biology of what I work on. Do we have time?
Yeah, I think the students would really appreciate that.
Great. Okay, so I work on a cellular pathway or in a cellular process that is called alternative RNA splicing. And I'm going to break this down. So we all know that we have our genetic material. It's our DNA that is packed into chromosomes. And basically this genetic material, it's composed of different entities. So it has exons, which are these colorful boxes. And these are the ones that carry the meaningful information to make proteins. And then we have these light blue boxes, which are called the introns. And the introns, we're still trying to understand really what they do, but most of them are kind of like silence material. They're there to provide kind of support for these exons, but at this moment, we don't understand very much what is their function. So after, so usually this DNA, it gets first transcribed into another molecule that is called the primary RNA. So pretty much the RNA, the primary RNA, is a single-stranded copy of this DNA. And you can still see that you have still exons, the colorful boxes, and you have the introns still placed here. Now, there is a need in this primary RNA to get rid of these introns because, as I mentioned before, they don't carry a lot of the genetic information. And so basically what the cell does is to remove these introns by this process of RNA splicing. It's going to take out all of those blue introns, and it's going to generate different types of proteins based on the different combinations of these exons. So for example, in here you have a mature mRNA, a mature RNA, sorry, that has different exons. It has exon 1, 2. Notice that you don't have the orange exon, and then you have purple, red, and blue. And this leads to the formation of one protein here, as mentioned. Now, you can have another scenario where you now are lacking exon 2 and exon 4, and you only have this RNA with 1, 3, 5, and 6. And it turns out that now, after translation, it gets formed another protein. And what we don't understand is why the cell produces such different arrays of proteins from the same DNA. And we're just starting to understand that sometimes these proteins may function similarly, but sometimes they don't. They function with very opposite functions. Now, on a third example here, you may have that the cell decided to take all the introns out and also other exons out. In this case, we're missing exon 2, and that's pretty much it. But the problem is that if you miss now exon 2, it turns out that this RNA is no longer stable and it will not form any protein. So this complexity, we're just trying to understand. Originally, we always thought a piece of DNA is going to form just one protein, right? 1 to 1. But now it's known that every gene or every kind of entity of the DNA actually can form up to 7 different proteins. And that's crazy because if you think about it, we were just talking about, the Avogadro number, which is a big number. So I'm going to give you another big number. There are about 95,000 genes or pieces of DNA that can encode proteins in our DNA. So 95,000. And if you multiply 95,000 times 7, because each one of them can form up to 7 different proteins on average, imagine how big that number would be. It would not be as large as the Avogadro number, but it still would be big. And each one of them would reflect on how complex and how plastic the genome is to produce so many proteins. And so that's what we are trying to do. We're trying to understand how cancer cells can produce one protein in one given scenario, or maybe it decides to turn and change and produce other protein to give them that flexibility of becoming so aggressive and growing tumors. So that's what my lab is trying to understand. We're trying to understand alternative splicing and how cancer cells use it to their advantage to grow and resist therapies. So with that, I just want to thank everyone, and please ask me any questions regarding the science that we do or my career path. Thank you.
Awesome. Thank you so much. Let's everyone give Luisa a big virtual hand. That was a really lovely talk. Thank you so much. All right. Lots of claps in the chat.
Thank you. Yes, I can see that.
I think the students, a couple of them had the same question, which I'll synthesize here,
Sure.
which can be boiled down to, what do you consider as sort of the most impactful discovery that, or, step forward even that you and your team have made?
Yeah, thank you for that question. Wow, that's a really nice question. So I have to say that the biggest discovery has been always how we come as a team to think about a problem in different ways. And it always surprises me how with the team we have different ideas that are really good ideas that can bring us to the lab to test them. But in particular, I wanted to talk about one discovery that we made last year, that we published last year. So it turns out that there are these two mutations that appear in tumors very commonly. One is a mutation in a gene called KRAS, and another one is a mutation in a gene called P53. So we've known for many, many years that several cancer types, not only pancreatic cancer, but lung cancer, ovarian cancer, always have the mutations on these two genes, but no one had ever studied how do they cooperate. The labs had studied just the mutation on KRAS, and they were like the experts on KRAS, and then the other group would only had studied P53, and they're the experts on P53. But what we found is actually how these two proteins cooperate to form tumors. And they actually do it because mutant P53 changes alternative splicing, that pathway that I was just showing you, and it changes in a way to support the activity of KRAS. And so that was a really fun discovery, because for the first time in 50 years since we discovered these mutations in these proteins, we now understand how do they cooperate. And what we found that is very interesting so that was kind of interesting from the kind of biology aspect. But now what we're doing is we are designing a drug that can actually block this cooperation so they can no longer function together, and we can actually better treat the tumors. So that's probably, I would say, what I feel right now very excited from the discovery, because it was ideas from different individuals that came together in the lab. And so it feels like a team effort, not like a solo effort. And the fact that we're going to try to bring this new therapy to the clinic between the Yale Cancer Center and Dartmouth College makes me feel excited, because potentially we can make a difference for the patients whose tumors have these two mutations.
Wow, that's very, very exciting to hear. I think that everyone can understand how impactful, that discovery was. So thank you so much for your time this morning. And let's, everyone give a final round of applause for Dr. Escobar-Hoyos.
An applause for every one of you. Thank you so much for the invitation, and I appreciate your time. And thank you to all the students, the parents, and the invitation. I really appreciate it.
Awesome.
Thank you.
Thank you.
Bye.
So we're going to now move into our next activity. And so for this, we are going to use our bags full of experimental materials. I know everyone's really excited about that.
And Caitlin, let me pause you for a second.
Yeah.
All the students, please put on your lab coats.
Yeah, so we're getting ready to do a little bit of science. So if you have your lab coats in your bags, get those things ready, put your lab coats on. What we're going to do is we're going to walk through 2 demonstrations. We're going to do the first demonstration. We're going to make our own stethoscopes, and then we're going to come back to the main room, talk a little bit about it, and then go back to do our second demonstration, which is building a paper circuit. And so you're going to be put into breakout rooms in a moment with 2 volunteers. And so you're going to introduce yourselves, get to know each other, and then move right into the activities. So what I'll do now is I'll have Heather put us into our breakout rooms so that we can get started on our experiments.